Let me introduce our last speaker, Professor Hans van der Vliet. Hans, whose work inspired Lava Therapeutics, and Hans will tell us about the use of T-cell engagers, specifically engaging Vγ9Vδ2 T cells in cancer immunotherapy. Hans?
Thank you, Andrea. Get the pointer. So thank you for the invitation, and so I will start with an introduction. May not be necessary anymore, but just to highlight the important role of Vγ9Vδ2 T cells in tumor immunity. This figure illustrates this. So Vγ9Vδ2 T cells and NK T cells that will become relevant later in the presentation can be positioned at the interface between innate and adaptive immunity. They are rapid responders and can interact via cytokines and in cell interactions with components of the innate immune system. And also Vγ9Vδ2 T cells have the unique ability to present antigens and thereby trigger naive CD4 and CD8 T cell responses. Vγ9Vδ2 T cells make up about 1 to 10% of all peripheral blood T cells and constitute a homogeneous, highly cytotoxic effector T cell population with the natural ability to recognize and kill tumor cells. So after the last speaker, I removed one figure in the left from a paper that everybody knows by now, but I'll show it anyway. So in 2015, a paper published by Gentles et al. In Nature Medicine illustrated the important positive prognostic role for gamma delta T cell infiltration in malignancies. And this was confirmed and elaborated upon by a study published 2 years later by Tosolini that confirmed the relative abundance of, in particular, Vγ9Vδ2 T cells in many different cancer types, ranging from hematologic as well as solid tumor indications in red and blue, respectively. So the Vγ9Vδ2 T cell receptor is a monomorphic T cell receptor. It interacts with a monomorphic antigen-presenting molecule from the butyrophilin class, as discussed before. So butyrophilin 2A1 and 3A1 can pair upon binding of phosphoantigens to an intracellular binding domain of BTN3A1, which results in a conformational alteration in butyrophilin 3A1, and this is sensed by the gamma delta T cell receptor. And these cells then become activated and can exert their pro-inflammatory effector functions. And via this pathway, cytolytic activity of Vγ9Vδ2 T cells against many different tumor types has been demonstrated. And because phosphoantigens are an intermediate of mevalonate metabolism, which is overactive typically in malignant cells, tumor cells typically have higher levels of these phosphoantigens and thereby are more susceptible to recognition and subsequent lysis by activated Vγ9Vδ2 T cells. So there have been earlier approaches to try to exploit this Vγ9Vδ2 T cell population for cancer immunotherapy. This is summarized on this slide. So earlier approaches either made use of ex vivo activation of expanded Vγ9Vδ2 T cells or in vivo activation approaches using either synthetic phosphoantigens like BRHBP or amino-bisphosphonate-based approaches, either alone or in combination with low-dose IL-2. And amino-bisphosphonates result in an upregulation of endogenous phosphoantigen levels and thereby trigger Vγ9Vδ2 T cell activity. So the important message of all these studies is that gamma delta T cell based therapeutic approaches appeared to be very safe. No signs of cytokine release syndrome were noted as a result of gamma delta T cell activation. And importantly, also some signs of clinical activity could be observed, as demonstrated in the top series of the CT scan, showing regression of renal cell carcinoma lung metastasis after adoptive cell therapy, And the bottom series illustrate regression of lymphoma lesions in a patient treated with aminobisphosphonate and low-dose IL-2. So despite the safety, the consistency and robustness of clinical responses was insufficient to take these approaches forward as such. And we think a major limitation in these approaches relies in the non-tumor targeted activation of these cells. So cells were either administered systemically or activated systemically. We believe that a tumor targeted approach, for example, using bispecific antibodies, could make this tumor targeted potential or exploit the anti-cancer potential of gamma delta T cells better, potentially resulting in a more consistent and robust anti-cancer response. So at LAVA, we make use of single domain antibodies or VHHs for the bispecific antibodies that we generate. So VHHs, single domain antibodies, are derived from a unique class of heavy chain only antibodies that are naturally present in camelids like llama, and the antigen binding region of a heavy chain only antibody has a size of about 15 kilodalton and has all the specificity required for antigen binding. So a bispecific single domain antibody could be created that has a size of 30 kD and contrasts with the size of a full regular IgG antibody. VHHs have several unique features that could be helpful. They are small, which may facilitate tumor penetration. They are very stable, can access unique epitopes, have an inherent low immunogenicity and good manufacturability. So we set out to immunize llamas with Vγ9Vδ2 T cells, and from this we used a phage display to select antibodies specific for either the delta chain or the gamma chain of these of human Vγ9Vδ2 antibodies, as illustrated here. So when these were then fused to, in this case, an antibody directed against EGFR, an EGFR gamma delta bispecific engager could be generated that resulted in potent lysis of tumor cells expressing EGFR and mediated by Vγ9Vδ2 T cell activation. This is illustrated in this slide. On the left, you see an EC50 of about 4 picomolar for killing of an EGFR expressing tumor cell line. And importantly, and in contrast to antibodies directed against EGFR that are currently in the clinic and are not effective against tumors that harbor a RES or BRAF mutation, our engagers can trigger gamma delta T cell activity again to tumors that express EGFR regardless of the presence of a RES or BRAF mutation. So SW480, HCT116 have a RES mutation, HT29 has a BRAF mutation. So some words on EGFR as a target. EGFR stands for epidermal growth factor receptor. It's a tyrosine kinase receptor, and ligand binding is known to induce signal transduction that is involved in cell proliferation, differentiation, and migration. And overexpression or aberrant signaling correlates with poor prognosis in cancer. And as mentioned on in the previous slide, anti-EGFR antibodies are effective and registered, for example, for metastatic colorectal cancer and head and neck squamous cell carcinoma, but activity is impaired in case of a RES or BRAF mutation. So to look in whether we could enhance the development of this of EGFR gamma delta engagers, of course, the presence of these cells would be required in patients. And indeed, Vγ9Vδ2 T cells are detected in circulation of patients with colorectal cancer at levels that are quite comparable to those observed in healthy donors, and also in tumor tissue of either the primary tumor, peritoneal metastasis of colorectal cancer, or liver metastasis of colorectal cancer. Vγ9Vδ2 T cells could be observed. And importantly, when we co-cultured colorectal cancer cells from peritoneal metastasis with autologous patient PBMC for 7 days. We could not only detect activation of the Vγ9Vδ2-T cell subset, but importantly also observed downstream activation of other immune cell components, including CD4 and CD8 T cells, as well as NK cells. We next explored whether the EGFR-γδ bispecific engager could also trigger Γ9Δ2 T cell activation of Γ9Δ2 T cells that are present in the tumor specimen of patients. So here you see induction of degranulation in the presence of the bispecific engager after a 4-hour culture, during a 4-hour culture of samples from patients with primary colorectal cancer, peritoneal metastasis of colorectal cancer, or liver metastasis of colorectal cancer. And importantly, in addition to the degranulation induction, the patient tumor-resident Γ9Δ2 T cells could also be triggered in an overnight assay to kill patient tumor cells as depicted here. So here, either primary colorectal or peritoneal metastatic lesions were dissociated, co-cultured with tumor-infiltrating T cell population as a whole. And as you can see, in the presence of the bispecific engager, lysis is clearly triggered. And also in co-cultures overnight of patient or PBMC and autologous tumor cells from peritoneal source. Again, the bispecific engager in an overnight assay triggered clear lysis of the tumor cell population. Importantly, and also observed by various by the previous speakers in this session, we confirmed the preferential activity of Γ9Δ2 T cells against tumor cells and demonstrate sparing of activity against normal cells. So here you see again that the bispecific engager can trigger Γ9Δ2 T cell activity to kill primary colorectal peritoneal lesions of colorectal cancer and liver metastasis, head and neck cancer lesions, and lung cancer lesions. In contrast, when we co-culture during the same conditions, patient-derived non-tumor tissue, so either normal colon tissue, normal peritoneum tissue, or normal liver tissue. I should add that this is from patients, but here we test the non-affected lesions. We see no lytic activity. So to further confirm or to explore the safety of this platform in non-human primates, we designed a surrogate molecule because the Γ9Δ2 T cell engager that we use for our human studies is not cross-reactive with the cyno TCR. So here, Vγ9 specific single chain Fv was fused to an EGFR specific single domain antibody binding to non-human primate and human Γ9Δ2 T cell receptor is similar. And also here you can see that the cross-reactive engager indeed also results in triggering of non-human primate Γ9Δ2 T cell degranulation. So the study was then set up using animals with consisting of 2 parts in the study, one a single dose study and another study where animals received 7 consecutive daily doses. And we started with 30 microgram per kilogram because this was a dose that was previously found to be fatal in combination with CD3 engagement of EGFR. So some data are demonstrated on this slide. So what we found after administration of this engager was binding of the engager to peripheral blood gamma delta T cells. We also found some activation of circulating gamma delta T cells. Of course, one cannot expect much activation in circulation because EGFR is typically not expressed in circulation. And importantly, also a low level cytokine spike with here IL-6 levels reaching a peak of 50 picograms per ml, which is very favorable compared to levels that have been previously reported by Luteboise et al., reaching close to 500,000 picograms per ml after administration of an EGFR-CD3 antibody. So what we noted was a temporary reduction of the gamma delta T cells in addition to the activation. And importantly, also in follow-up studies, we observed no clinical signs of toxicity, no clinical chemistry abnormalities, and no histopathological abnormalities. Then moving to CD1d as a tumor target. So CD1d is an MHC class 1 related glycoprotein that is known to present glycolipid antigens to natural killer T cells, as demonstrated here. CD1d is expressed on the surface of various human antigen presenting cells, but also expressed on various malignancies, in particular hematologic indications like CLL, AML, TALL, and multiple myeloma are known to express CD1d. And expression has also been noted on several other solid tumors. CD1d is also expressed by immunosuppressive cells in the tumor microenvironment, such as NDSC and tumor-associated macrophages. And previously, glycolipid antigen-based approaches and NKT cell adoptive cell therapy have confirmed a good safety profile for therapies targeting the CD1d iNKT cell axis. So we immunized llama with CD1d positive tumor cells, and then that resulted in the creation of a VHH panel with VHHs specific for CD1d and no cross-reactivity to CD1c, CD1b, or CD1a. When we looked at the functional consequences that these VHHs had, we found multiple CD1d specific VHHs that had no relevant effect on natural killer T cells that are, as I mentioned before, known to normally interact with CD1d. One VHH, 1D22, was found to block NKT cell activation, as illustrated here. And an important observation was that one VHH termed 1D12 had the unique ability to result in activation of natural killer T cells. And a crystal structure of that binding of that VHH is demonstrated here. So the CD1d specific VHH 1D12 had the unique ability to trigger NKT cell activation, and we found that this was due to intrinsic bispecificity of this VHH. So as you can see here, CD1d can interact with the NKT T cell receptor, and the VHH fits perfectly in the groove between the two receptors. The top view of CD1d shows the footprints of the VHH and the TCR alpha and beta chain of the NKT cell receptor, and the side view of the T cell receptor alpha chain and CD1d illustrate various binding or the footprint of the VHH on both CD1d as well as the NKT T cell receptor. And as encircled here, this VHH interacts thereby not only with CD1d but also with the NKT T cell receptor. And if we replace arginine on these positions by an alanine, we can actually abolish the intrinsic bispecificity and the NKT cell activating potential of this VHH. So when we next fuse the CD1d specific VHH 1D12 to a Vδ2 specific single domain antibody, we create a bispecific single domain antibody that has the potential to stimulate both type 1 NKT cells as well as gamma delta T cells. So it engages two immune effector cell populations. And this is illustrated here for degranulation using CD1d positive myeloma cells or CD1d positive TALL cells as target cells to see activation and degranulation of, in red, gamma delta T cells and in blue NKT cells. And this also results in cytotoxicity of tumor cells expressing CD1d This can be mediated by either gamma delta T cells or NKT cells. And the EC50 for gamma delta T cells is in the single-digit picomolar range. For NKT cells, it's in the sub-nanomolar range. LAVA51, which is the bispecific single-domain antibody that targets CD1d and the gamma delta T cell receptor, can also trigger cytokine production in co-cultures of tumor cells and effector cells, and either NKT or gamma delta or the combination thereof can trigger cytokine production like IL-2, 4, TNF, and interferon gamma. Importantly, it can also trigger expansion of the NKT and gamma delta T cell population in co-cultures of PBMC and tumor cells. And again, to confirm earlier data, we also, with this engager, see preferential activity against tumor cells expressing CD1d, like here, with minimal to no activity against normal cells expressing CD1d. So CD1d is expressed by tumor cells of patients with CLL, MM, and AML, as depicted here. You see individual patient data for CD1d expression on CLL, multiple myeloma, and AML. And noteworthy is that in AML, CD1d is predominantly expressed by the myelomonocytic and monocytic subtypes of disease, M4 and 5. The engager can also be used to trigger cytotoxicity against patient cells from either CLL, MM, or AML, as depicted here. So in red, the induction of T-granulation of patient gamma delta T cells against in co-cultures of the tumor cell population, and the right side of this slide shows data of cytotoxic activity of gamma-9 delta 2T and NKT cells against patient CLL, MM, or AML cells. Several in vivo studies have been performed to look whether we could also find an in vivo anti-cancer effect of this approach. So here on the left, you see a model in immunodeficient mice where CD1d positive myeloma cells were infused on day 0, and treatment was started from day 7, consisting of either the bispecific alone, NKT and gamma delta T cells alone, or in combination with the engager. And strong anti-tumor activity could be triggered by the bispecific engager, as seen here, translating into improved survival. And also in the subcutaneous TALL model, where TALL cells were co-implanted with PBMC, so non-enriched, non-pre-activated gamma delta T cells, we could see tumor growth delay and improved survival when the bispecific was administered to these mice biweekly during the first 14 days. So to also explore the safety of targeting CD1d in non-human primates, we designed 2 cross-reactive engagers, one termed LAVA39, which is fully cross-reactive, so it binds to the gamma delta T cell receptor of non-human primates, but also to CD1d of non-human primates, and LAVA48, which only binds to the gamma delta T cell receptor and does not cross-react with CD1d. And again, these compounds were explored in Macaca fascicularis, a single-dose study with 3 different doses, and a follow-up study where 7 daily doses were given to non-human primates. The figures below illustrate again that the 39 compound binds to CD1d expressing monocytes in non-human primates, to gamma delta T cells in non-human primates, and can result in activation of gamma delta and degranulation of gamma delta T cells in non-human primates. When we then look at pharmacodynamic markers, we see that both LAVA39, so the dual cross-reactive engager, and 48 can bind to gamma-9 delta 2 T cells in circulation, and this can be detected still after a few days. Only the cross-reactive engager also results in a temporary decrease of gamma delta T cells, quite similar to observations that were reported by Daniel Aliv in the clinical study. No such decrease is observed in with LAVA-48, and importantly also signs of gamma delta T cell activation after administration of the cross-reactive engager were observed, as noted here. Cytokine levels again were quite favorable, low cytokine spike after the first administration of the engager. And importantly, also in these studies, we saw no clinical signs of toxicity, no clinical chemistry abnormalities, no histopathological abnormalities, and also important, no depletion of normal CD1d-expressing B cells and monocytes. So at LAVA, we developed two bispecific gamma delta T cell engaging formats, a bispecific single domain antibody with a high affinity, high potency, an expected short in vivo half-life based on the small size of the construct, but is prolonged functionally because of high affinity binding to the T cell receptor and gamma delta T cells in circulation. It's a small molecule, size of 30 kD, and this is used for our lead hematologic program, LAVA-51. The other format consists of a bispecific single domain antibody with an Fc domain connected to it to increase the in vivo half-life. This molecule still is fairly small. It has a size of about 80 kD, which is half the size of conventional antibody, and the Fc domain has validated mutations to silence Fc effector functions. And this format is used for our lead solid tumor program, LAVA-1207, in metastatic castrate-resistant prostate cancer. So the LAVA-51 first-in-human phase 1/2a study design is shown here. So we have dose escalation part in patients with CLL and MM, and somewhat later in the study we also include patients with AML. And then those expansion cohorts are available for the phase 2 part, where we look at CLL, MM, and AML expansion cohorts. We perform extensive monitoring during the study, looking, of course, at safety, disease assessments, pharmacokinetics, and anti-drug antibody development. And focusing on immune readouts, we look at cytokines, binding of LAVA-51 to gamma delta T and tumor cells that express CD1d, the activation status of both gamma delta as well as NKT cells, and we also look at, of course, at monitoring we monitor conventional NK and T cell subsets and others. So our pipeline crosses hematologic and solid malignancies. So LAVA-51 is a trial that is currently recruiting. LAVA-1207 is targeted against PSMA in MCRPC, CTA and IND approved, and the trial will be open for recruitment within the next week or so. And then we have follow-up programs, LAVA-1223 directed against EGFR, CD40 preclinical program, and the biotech collaboration with Janssen that is ongoing. So in conclusion, we think there's a strong scientific and clinical rationale for tumor-targeted engagement of Gamma-9 delta 2 T cells. We have a panel of agonistic camelid single-domain antibodies available that can be combined with tumor-associated antigen-specific antibodies in the bispecific approach. It's a modular platform, and the use of camelid VHHs offers multiple upsides like low immunogenicity, small size facilitating tumor penetration, and importantly, we also differentiate from a pan T cell approach because on the one hand, the gamma delta T cell restricted activation can be expected to result in a more benign safety profile. And importantly, in contrast to CD3 T cell engagers that trigger activity of all T cells, including suppressor T cell populations that have also in clinical studies been shown to have a dampening effect on the anti-tumor response, we selectively engage Vγ9Vδ2 T cells and avoid co-activation of suppressor T cell populations. The safety of our platform has been demonstrated with surrogate engagers in multiple non-human primate studies. And as mentioned, we have two clinical trials open for recruitment, or actually one open for recruitment and the next within about 1 week open for recruitment. And I'd like to acknowledge people, especially Roland Lameres, Lisa King from Amsterdam UMC, collaborations with Melbourne University, Monash University, and many people. This is not a complete list of course, at Lava Therapeutics. Thank you.